This lecture will be on Chapter 20, Hematology Automation and Flow Cell Cytometry. At the end of this lecture, the student will be able to describe the principle of electrical impedance, summarize how histograms and scatter plots assist in automated data reporting, define the routine quality control procedures used in automated methods that ensure valid patient results, identify the manner in which flagging criteria, delta checks, and reflex testing assist the operator in providing quality patient results. Define the function of the reticulocyte hemoglobin concentration parameter and the automated instruments that use this parameter. List 3 applications of flow cytometry. Define cluster of difference and describe the difference in flow cytometry between a contour plot and a single or two parameter histogram. When it comes to hematology automation or hematology instruments, there are different ways that the results are determined. The first one we're going to talk about is electrical impedance. The cells are sized and counted by detecting and measuring changes in electrical resistance when a particle passes through a small aperture. 2 electrodes establish an electrical current, and the external electrode is located in the blood suspension, which is the blood sample from the patient diluted in saline, and there are internal electrodes that contain this aperture that it passes through. Here you can see a diagram of what the previous slide described. So here you can see the external electrode, the internal electrode here. You have the aperture bath, which is the sample or the blood cell solution, which is the patient sample mixed with saline. And the sample runs through this electrical current, and that's how it's able to count the cells and determine what the cells are due to the electrical impedance. Another thing that the hematology analyzer offers are histograms. And this is a graphic representation of the cell frequencies versus the size. The pulses are channelized by the height, and the data is collected and plotted. The relative number is on the y-axis, and the size or volume is on the x-axis. This provides information about the red cells, the white cells, and even the platelet frequency. The distribution about the mean of the total number of cells and the presence of subpopulations of cells. This is some graphic examples of histograms in particular from a culture. That's the brand name of a hematology analyzer. This top image shows a histogram of leukocytes. So you can see we have the bottom number is the quantification, the femtoliters, and the relative number is on the left side here. You can see how the lymphocytes plot. Mononuclear cells are monocytes, and then your granulocytes. This diagram marked B shows the histogram for your red blood cells, and C is the histogram for your platelets. We also have the use of radio frequency when it comes to hematology automation or hematology analyzers. There is a high voltage electromagnetic current. It detects the cell size based on the cellular density. The cell interior density is directly proportional to the pulse size. So the nuclear to cytoplasm ratio, which we talked about previously, is determined. The nuclear density and cytoplasmic granulation are also determined. When it comes to the opacity, that's actually derived from a mathematical equation. So the opacity is a mathematical equation that's derived from the ratio of the radio frequency, or RF, and the DC. The cells with a higher nucleus to cytoplasm ratio have a low opacity, and cells with a lower nucleus to cytoplasm ratio have a higher opacity. We also have scatter plots, which is a graphic representation of two or more measurable characteristics of a cell. It uses conductivity and light scatter to visualize and analyze the cell data. And this image is from a particular instrument, the Beckman Coulter, one of the Beckman Coulter CBC analyzers, and this shows a three-dimensional scattergram, and you can see each area. This blue area is the lymphocytes, the green area is the monocytes, your neutrophils, this pink area, and your eos in this orange area. An optical scatter is when the cells pass through a flow cell on which a beam of light is focused. The flow cells are composed of quartz rather than glass and provide a better measurement of cellular qualities. As the cells pass through the sensing zone, light scatters. Photodetectors sense and collect scattered rays and convert that data into an electrical pulse. So the number of pulses is directly proportional to the number of cells passing through the sensing zone. We also have VCS technology, which stands for volume, conductivity, and laser light scatter. This differentiates white blood cell characteristics. You have hydrodynamic focusing, which is a technique that narrows the stream of cells to a single file. It allows for greater accuracy and resolution of the blood cells. It eliminates recirculation of cells and the necessity for counting the cells twice. Flow cytometry uses a laser light source to measure the physical or antigenic characteristics of cells as they pass through the beam of the laser light. It can evaluate nuclear DNA content, surface antigens, and cytoplasmic antigens. The light scatter signals are amplified. It's converted to digital signals, and it's plotted to a scattergram that separates cells into cell clusters. Cells of interest can be selected for immunophenotyping and DNA content analysis. Here we have an example of a two-dimensional scattergram, and you can see the black area here is our neutrophils, blue is our monos, red is our lymphocytes, and it doesn't say here, but this green area is most likely your basophils. Within flow cytometry, you also have falls or forward light scatter, and this evaluates the cell size. You have SSC or side scatter. This evaluates the cytoplasmic and nuclear complexity. You also have MALS, which is median angle light scatter. This is proportional to the cell size, the granularity, the surface properties, and the reflectance. Flow cytometry, you can have multiple angle polarized scatter separation. So there's 4 degrees of separation. 90 degrees measures the lobularity. 90 degrees D measures the granularity. 10 degrees measures complexity, and 0 degrees measures the size. These measurements identify the 5 basic subpopulation of cells, as we mentioned at the previous slides. So this gives you a diagram of what we just talked about, of the different degrees and how that works under maps. And you can see the angles of like 90 degrees is the lobularity, 90 degrees D is the granularity, 10 degrees complexity, it's cutting straight through there, you watch if you follow that purple line, 0 degrees, which is completely horizontal, cuts straight through here, and you have the 4 degrees of separation in a flash. Optical platelet counting and flow cytometry, you have two-dimensional analysis of low angle and high angle light scatter. The scatter measurements are related to the platelet size or volume and the refractive index or the density. There's a patented fluorescence dye that's used. The blood sample is coated with CD61 antibody, and then you use the flow cytometry with high forward angle and light scatter. So when it comes to the instruments, you have automated hematology analyzers, and they measure your red blood cells, your white blood cells, your platelet count, your mean platelet volume, your hemoglobin, and your mean red cell volume. There are also things that the instrument will calculate. It's not directly quantified. It's a calculation, and that would be our hematocrit, our mean cell hemoglobin, our mean cell hemoglobin concentration, as well as our red blood cell distribution width. Newer analyzers are typically all analyzers at this point. You will get a white blood cell differential, relative and absolute white blood cell number, which we talked about in the previous lecture, a reticulocyte analysis. The newer generation of instrumentation also offers enumeration of nucleated red blood cells, which are immature red blood cells. Verification system reviews past patient results, uses delta checks and instrument flagging. That's pretty standard. You do need to be aware of, when a patient has abnormal results, to be in the habit of going back and looking at previous results if you have any, to do your deltas, as well as being aware of any parameters the instrument's going to flag and what you do with those flags. So with the report that you get from the hematology analyzer, you're going to get numerical values. You're going to get graphics that are displayed, whether that's scatter plots, scattergrams, or histograms. Now these next slides, actually slide 23 through 55, is going to break down specific instrumentation that you may or may not see once you get to your clinical rotation and eventually your jobs. I did not want to cut this information. I encourage you to read through it. You can have this available to you if you want to. When you get to the point where you're starting your clinicals, you can go back and go, oh, okay, my site has a Beckman Coulter instrument in hematology. I want to brush up on how this instrument works. So I did not want to cut it all together. However, if I go through and read everything specifically, it's not really going to click with you guys yet because you're not quite there. You haven't laid your hands on any type of major instrumentation. So I'm going to work ahead. I'm going to cut this here, and I'm going to pick up at after 55 on what I can elaborate on that's more clinically relevant for you guys at this point. So I was working through those slides. I tried to stop for a few seconds on each slide, getting to the point that we were past all the specific instrumentation. That way, if you're watching it and you wanted to pause it at that point to read it, you could. So now we're going to talk about quality assurance and quality control. So the CLSI, which is the Clinical and Laboratory Standards Institute, provides guidelines for quality assurance and quality control. In particular, we have instrument calibration. You want to do that with the initial installation of a new instrument, and it should be done at least every 6 months. Calibration verification. You want to do this after any type of instrument repair or replacing anything on that instrument. You also have quality control, which ensures your accuracy and precision of your results to make sure you are providing the patient with accurate results as far as the best quality care that you can provide. So quality control, so when it comes to how we use quality control in the laboratory, we establish or verify the manufacturer means before using the control material. You typically have 3 levels of control that are ran every 8 hours or every shift before you can report out any patient testing, and because you want to verify your testing method is working properly. And the acceptability of how your controls are running should be evaluated daily. Lastly, we're going to talk about flow cytometry instrumentation. This measures physical characteristics, chemical properties, and phenotype of cells, and it defines the maturation of the cell stages. It uses light scatter, light excitation, and emission of fluorochrome to generate specific data from cells or particles. It's used in the aid for diagnosis, prognosis, and the monitoring of disease states. The principles of operation behind flow cytometry consists of 3 systems. You have fluidics, or the transport system, optics for the detection of light scatter and fluorescent signals, electronics to monitor and control the operation and the storage of that data, and it also uses hydrodynamic focusing. The fluidic system is a pumping system that transports cells from a sample suspension to a flow chamber at a very high speed. The hydrodynamic properties causes the cells to line up in a single file line for excitation by the laser. Then you also have the optics system, which provides illumination of stained or unstained particles, and it detects the light scatter and fluorescent light signals. Every cell has a unique fluorescent profile or scatter pattern. The information from the light scatter and emission from the fluorescent light is separated and directed to a photomultiplier tubes, or PMTs. We also have the electronics, which is tied to the control, acquisition, analysis, and storage of this data. The light signals are collected by the PMT directors, and those are converted to digital signals by the system's computer. The results received from the flow cytometry are quantitative in absolute numbers and in the fluorescence intensity. The analysis comes from the determining of the light scattering properties of the cells, and the higher the falls, which is the forward angle light scatter, the larger the cell. The greater the side angle light scatter signal, the more internally complex is the cell. So more mature monocytes, lymphocytes, and granulocytes are identified through their light scattering properties. The next process is called gating, which is a population of cells is identified by further analysis by the fluorescence. So the fluorochrome is attached to a monoclonal antibody. The cluster of differentiation, which is also known as CD, you'll see that abbreviation CD, is the cluster of antibodies that is recognizing the same antigen that is on the cell. The monoclonal antibody markers are used, each labeled with a different fluorochrome. The fluorescent data can be examined for absolute number or percentage of cells that are positive and fluorescent intensity. The data can be displayed in a single parameter or two parameter histogram. There's a dot plot that's displayed for each cell on the graph as a single event, and there's a two parameter plots that are divided into two different quadrants. The percentage of positive cells are calculated based on the number of events within each quadrant. So you have the x-axis and your y-axis here. Within flow cytometry instrumentation, you have quality control. You have internal quality control, which is checking the performance of the lasers, the PMTs, the optical filters, and the amplifiers. And then you have normal controls that must be tested daily, as we mentioned previously. And then you have external QC, which is by running distributed control samples that you can get from the manufacturer and comparing the performance of one laboratory with the performance of others. When it comes to flow cytometry instrumentation, it can be used in the diagnosis of leukemia and lymphoma, of immunodeficiency disorders, to evaluate the neutrophil function to aid in the diagnosis of CGD, the detection of HLA alloantibody. It can enumerate the CD34 stem and progenitor cells in organ transplantation, and it also can be used in transfusion medicine.